Hey everybody, thanks so much for joining today. Um, yeah. So today we have a great presentation for you given to us by Alexis, Dr. Alexis Paez. She's going to be doing a webinar aimed towards VSS. And if you didn't know, there is a virtual VSS going on right now. It normally runs in May. It got pushed back because of the pandemic. If you've never been, it's a great place to, it's a great conference. There's lots of extra things to do. It's right on the beach in Florida every year. I've been a couple times. NeuroNexus has sponsored for multiple years. I don't know how many, but I would definitely recommend checking that out sometime. And I also wanted to mention that next week, next Thursday would be July 2nd. So we're going to take that week off so that people might be going away for the 4th of July holiday. So we'll just take that one off. And then coming up in July, we have some great webinars planned. We have a matrix planned. We also have a Lego updates. We have many updates from a Lego. It's our software for the smart box, but you can also use it without the smart box for spike sorting, and we'll have people come in and tell you all about all the features that you can use now. And we're also going to have a special guest in July, which I'm not going to mention just yet. So with that, I'm going to say, take it away, Alexis.
Hi, everyone. Good morning to the US, good day to Europe. Thanks for being here. As Matt said, we did sponsor VVSS this year. We have a fancy video ad up on the exhibitor page. If you want to view that, it'll just remind you to come back and contact us, which you've already done if you're in touch with us now. And it did, I think, end yesterday, but that video will be up for a few months. So thank you to all vision scientists who have joined us, maybe haven't joined us before, and everyone else who's interested in vision science. Of course, contact us or mention in the chat or wherever you want. If you are partial to another system, I studied the vestibular system and the oculomotor system, which ties to the visual system. Let us know and we can do another one of these. It seems like a lot of fun, at least from my perspective. I hope it'll be fun for yours. So here we go. So my goal today is to present some recent publications in vision science, and all of these papers, there are 4 here, are available online. And at the same time I present the actual publications, I'm going to, of course, overview the NeuroNexus products that were cited in them. And I have 2 main topics in the talk. So one is about the development of complex cells. So a fun part of the visual system. I know back in the day, way at the beginning when Hubel and Wiesel started with simple cells, now we know more about complex cells and even extremely complex cells. I forget the word, what they're called now. But 2 papers for that, as then another set of 2 papers with the recently hot topic of behavioral testing in rodents. When I was learning about how popular this is now, I worked with macaques, and now knowing that mice got a lot smarter. Matt just put a link, by the way. I checked the chat to make sure that wasn't somebody saying that I disappeared or something. These 2 papers talk about behavioral testing involving behavioral tests in rodents. And then, of course, I'll mention a lot of these are cortical recordings. We also have our products used elsewhere in the visual system. And then, of course, we'll take the live questions as promised at the end. So the first publication that I want to speak of is titled "The Neurons That Mistook a Hat for a Face." And I loved that when I saw it because I have read the Oliver Sacks book, The Man Who Mistook His Wife for a Hat. And this paper, this presentation is just as fun as it sounds. I got to see it presented as a poster at VSS last year. And I wish to congratulate the group from Harvard Medical School, Marge Livingstone's lab, who recently published the paper. None of my controls are behaving. Let's see, advance the slide while I'll use my mouse. Okay, So the premise of this is that a lot of visual studies, as you all know, study specific cell types and what they encode. So face neurons, if you present a face into their receptive field, their firing rate goes up. If you present a house in the receptive field of a place neuron, their firing rate goes up. But then there's also the fact that object recognition requires some sort of perception, some sort of integration of the context of the thing. So if you see an object out of context, it's harder to recognize what exactly it is. Some little stats, and I will give this for each of the publications that I'm citing. This experiment was done with adult macaques in the infratemporal cortex. They did chronic recordings using the NeuroNexus matrix array. I'm just going to open up a quick note so I don't forget. I had some questions this morning that I wanted to add in. So what we already know about face neurons in infratemporal cortex is that if you present an image that has faces, so what these authors did is take an image like this, present it at a bunch of different locations so that each part of this image eventually showed up in the receptive field of each neuron. And then this is a population composite of neuronal firing rates, so change in neuronal firing rate when a given part of the image showed up in the receptive field. And what these three results are showing from two different monkeys is that these neurons are happy to see faces. What this study also demonstrated is that these neurons also respond to areas in the visual field where there ought to be a face. So these neurons in this example are responding to occluded faces. So there's a rug here, but it's a rug on top of shoulders and a torso. There's a box here, but it's a box on top of shoulders and a torso. There's a box here, but it's a box held in above arms holding a different object, etc. So what these plots on the right are showing, the dotted lines show where the firing rate change is significant. And so at this interval after stimulus onset, faces, of course, elicit the largest response, but even occluded faces elicit a significantly different response from non-faces, like any other part of the image. Likewise, these neurons are responding to masked faces. So when there's not just a face sort of blocked, so you can see there's still a head here, still some hair, there's still some hair. Here, there's a total covering, like a bag or a mask or something. And I think this is fortunate that we do all still recognize each other as human or primate, in spite of everyone wearing masks and face shields lately. And this is why. You can thank your neurons for recognizing that. So masked faces, the same thing. The brain or your infratemporal cortex is still encoding, responding strongly to where the face ought to be. Whereas, so this is where you get the context clues. If I saw, or if we were presented with this paper bag or this shield or something else in isolation, these neurons would not respond to that. So that's what this study is pointing out. And finally, here's the image where the neurons are mistaking a hat for a face. You can see that once this gentleman's hat is transposed up onto his shoulders and body, the neurons think that it's a face. Less so with the bowling ball. When this little boy's hands are put where his face should be, the neurons think it's a face. When this man's face is put down on the ground, of course, it's still a face. It's recognized as a face. But when his shirt logo is moved up, it's almost as exciting as the monkey's face. Hand on the head becomes something that should be a face in context. And then the other half of this paper, which I'm gonna rush through, is that they also in this study presented like a similar series of beautiful images and results, recordings, where they probed the orientation encoding of the face on the body. So that's the context, right? So they were probing the actual context of what should be a face, or when there is a face or something that represents a face on a shoulders and torso and a body. If it's upside down, does it still recognize? Is it any sort of, is there an orientation selectivity to the context as well? This is the probe that this lab used. So the fun part about IT cortexes is sort of off to the side. So it's hard to maybe implant a large set of connectors for 128 channels on the side of the head. So what we have is a hybrid matrix array. And the hybrid means there's a flexible cable extending from the actual connector package and housing to your platform. So they can implant the platform in their craniotomy, run the cable to somewhere more stable or on the top of the head, et cetera, where they can implant or fix this casing. So the M is for matrix, the H is for hybrid, the S is for strengthened. So we have a small animal matrix that would look like this, but it's plastic. This is a strengthened version. It's a metal version that has either these feet you can cement on, or it could have larger feet with holes that you could drill onto the skull, etc. When you remove, so this cap is watertight. So there'd be a gasket and set screws that hold it on, so that when your animal is back in its cage, it's not going to get gunked up and ruin your experiment. When you remove the cap, this one is configured to connect to two 64-channel head stages. And the actual probe in this platform is what we call the M4x8. So these are specific to the matrix array. The platform would have 4 of this aligned inside of it to get you 128 channels. So it's 4 shanks each with 8 channels on them, 8 electrode sites on it, times 4 gets you that 128 channel 3D volume of recording. So what we've learned from this study is that these complex cells that know a face also integrate something about the context. So it's not just those objects without context. The responses actually take into account likely experience. So that's unsupervised spatial learning is the term you could use, where you're not sat and trained when you're growing up to say a head belongs on shoulders, belongs on a torso, belongs on legs, and then there's usually feet at the bottom. You just know that. You're used to seeing it. And if you saw a disembodied torso, it'd be a little strange. If you saw a head floating in space, that'd be a little strange. So that's what the previous study looked at, was unsupervised spatial learning. And then the next paper that I'd like to present deals with unsupervised temporal learning. So this next study deals with developing rats. They recorded in primary visual cortex, did acute recordings with these 2 designs, which I'll talk about in a moment. But the paper is called Unsupervised Experience with Temporal Continuity of the Visual Environment is causally involved in the development of V1 complex cells. So this is talking about how we get those cells that have responses to more complicated things than just, hey, there's an object in my visual field. And the way that this experiment worked was that pups were reared in the dark when their eyes open at P14, 2 weeks after birth. They were kept in the dark most of the time, and their visual experience was videos. So certain length videos, a couple minutes to half an hour, I believe was the longest one. A control group was shown natural videos that played in order. So hey, this rat, if it's walking, you would see its head followed by its tail, the body followed by its tail, or if a ball is dropped, it's going to go from the top to the bottom of the image, something like that. And then the experimental group was shown the same videos, but the frames of the video were randomly shuffled. So the other interesting part is that each frame was shown long enough so that the brain could perceive the spatial characteristics of each frame, but then the temporal relationships within the video were completely lost. And then after P60, so when the pups are 2 months old, the study started acute recordings in V1. And these are the probes that were used in those recordings. So some were 32 channel, they would be on this connector here. 32 channel probe, 5 millimeters long, and the sites are spaced 25 microns apart. So this is one of the closest spacings that we offer, and it provides a very dense recording. But the linear design also allows sampling from all the layers of cortex. You can reuse and reuse and reuse these probes, which is what was done. You can also reuse our more chronic probes, but the advantage of this, you wouldn't want to implant this probe because it's very large. You can see it's 5 centimeters long. But the advantage of this is it's very easy to handle, and it's a little bit less expensive. And then the lab also expanded, so they took the exact same design. So it's a 5 millimeter long with 25 micron spacing, this 177 square micron electrode site, and basically doubled it. Now it's a 2-shank probe. This 200 is the shank spacing, and they're using the 64 channel acute connector. So the scale is a little bit different. This same grid of pins would be one of these, and here's a second one, to get 64 channels. So what they did was they didn't sacrifice any span, they didn't sacrifice any density, but what they did was double their recording. So if you have the capability to scale up your connector, then this is an option. And even if we don't offer something like that in the catalog, just ask us, and it's very easy to, especially with something we've already made in one shank, to make it 2 shanks is probably very easy, unless you want the 2 shanks, centimeters apart, probably won't be as easy. Anyways, this paper, so another thing about these acute probes, this particular lab developed a method for identifying where the probe had been inserted in terms of depth based on the visually evoked potentials that they recorded. So the VEPs with a template matching algorithm allowed them to estimate, and the point here is that in the control animals and in the experimental animals, the experimenters recorded from the same, approximately the same, basically the same depth in the brain. They were targeting layer 5, got some of layer 4. And the neurons that they recorded, oh, I will say also, we recommend as alternatives, if you're not familiar with this method, and I wasn't until this week, we also recommend you can sacrifice a couple of recording sites, say maybe the top one and the bottom one, to make electrolytic lesions, and then you can reuse the probe again and record from the middle 30 sites, for example. Or you can paint the back of the shank very, very gently with something like dye eye, which will mark your track as well. So we have a technical note on the microlesioning, and I think we do have some resources on using some dyes as well. In this paper, when they recorded from approximately layer 5, some layer 4, they did record both complex cells and simple cells. So here's just a canonical example. This is with their example cells. The complex cell does have orientation tuning, but no real phase response, whereas the simple cell, which has the more center surround receptive field, has the orientation tuning and is very phasic in its response across. So I neglected to say, during these tests, during the recordings, the animals were shown a moving gradient. And what the results showed, based on this modulation index, the phasic nature of the responses, the complex cells would have a lower modulation index, like shown over here. The simple cells would have a higher modulation index, shown over here. And what they found is that there was a significantly greater proportion of complex cells in the control group than in the experimental group. And what that led them to believe is that, or to conclude, is that the complex cells were not developing as much when the temporal aspect of context was not present in the visual environment. So that concludes this part, the complex cell discussion. And I wanted to jump to visual system recordings during behaviors. So we're all pretty familiar, I think, with having an animal restrained and having some sort of screen in front of them. These previous two, they were passively viewing stimuli. In this study and the one following, the animals were actually asked to perform a task. So in this paper, the animal model is a mouse. Again, the recording target is primary visual cortex. The experiments were daily acute, so there were acute recordings on some successive days for several days. And they used 2 different probes. Slightly different story from the previous one, and I'll talk about that in a minute in terms of the choice of product. And a paper is called Spatial Attention Enhances Network Cellular and Subthreshold Responses in Mouse Visual Cortex. So in this paper, the mice were trained on a simple detection task, as I mentioned, and in their task, the stimulus was either monocularly presented or binocularly presented, and the trials were grouped in blocks of either a whole bunch of trials, I think it was 25, with a monocular stimulus switch, and there'd be 25 in a row. The binocular stimulus, or vice versa, recordings in B1, and the mouse has to lick within a certain response window after the stimulus is presented. The V1 recordings, again, I said I'd explain the choice of product here. Again, it's a 32-channel linear probe, but this one's actually our edge design. Some people prefer this because the sites are right up to the edge. You might get a slightly different sampling than when the sites are in the center. And it also affords, because these sites are more rectangular than circular. There's a slightly, I told you 25 microns was our smallest, one of our smallest. The 20 microns here is the smallest. So this is a very dense, but functionally linear array. And then this group chose another, so the A32 is that connector I had shown you before. This other probe is also an A32 probe, so it's another 32 channel probe, but they chose to spread the channels out over 2 shanks. So it's a 2 by 16 channel probe, same length, double roughly the spacing. So the span here is not changed so much, it's around 700 plus or minus on both, but now you have some horizontal span as well. And so I think the 50 micron spacing is still close enough to get sampling across the layers of V1 and some redundancy in your recordings. And these two shanks are 500 microns apart, so farther apart than the 200 that we saw on the matrix, for example. So it's just a way to expand your recording if you want to record from different cortical columns, if you want to record from different regions, things like that. And in this study, okay, so the reason that this trials, by having the trials blocked into all monocular, then all binocular, and then switch, the premise was that once the mouse settles in, knowing that the stimuli are going to be all the same for the next several trials, their reaction time goes down and their performance goes up. And the authors did test for, arousal, like being present in the task, not just getting better at it for other reasons. And likewise, when behavior gets better, the neurons show a change as well. They're modulated more. So for example, both for the inhibitory neurons and the excitatory neurons, the fire mates you can see from early trials, there's not as much of a depth. And then the later trials, so in all of these, the bolder color is the later trials. And when the stimulus is inside the receptive field, you get the greatest difference from early trials to later trials. And here I'll just highlight panel B, shows the LFP. So this study was also able to add patch clamp recordings to see if the change in the LFP from early to later and beyond trials was due to subthreshold membrane potentials. And another advantage, so I'll just sum up here, the exciting part about having behavioral tests available in rodents. So this study said it was a novelty to translate a primate behavior test into rodents. The exciting part about doing that is that you can integrate more methods. So for example, the patch clamp I know would be very hard to do in a primate because even though they're head restrained, they wouldn't be anesthetized, they could always move, it'd be very hard to hold a cell. Likewise, optogenetics. So they did include that as well. Of course, that's why it's in a Nature, a Nature journal. So it's very easy to include optogenetics in a rodent, more so than in a primate still. And there is a need to understand more of the circuit than just what's going on in V1. And so in rodents, it seems that there is that capability more so than in primates where things are just closer together. So to speak to that, I wanted to say this fourth publication in the talk, Movement and Performance Explain Widespread Cortical Activity in a Visual Detection Task. This is another visual detection task in a mouse. The target recording area was actually more cortex. The probe was in V1. Again, acute recordings. This time a 16-channel probe, but it's a polytrode, so I'll mention that. So just a brief background, the mice were trained, again, on a visual detection task. Their electrophysiology targeted specifically layer 5 of visual cortex. And what this study was able to add was wide field imaging to the larger region of the brain. So let me speak to this layer 5 recording. This lab used a polytrode. This is another layout that NeuroNexus offers. So I showed you edge and I showed you linear. This is a polytrode, which means it's like a linear probe, but sort of doubled and staggered. And it's also kind of like multiple tetrodes stacked together. So it's an advantage of dense recording as well as some span. So in this case, because there's just 16 channels, they have a relatively short recording span and relatively dense. So in this case, each site is 50 microns away from the next, just like the previous one. So it's still redundant. And the short recording span means most of the sites are isolated to a layer and they targeted layer 5. And the main result from this paper, I cannot show you the figure. I did say these were all open access, but this paper is readily available online and not open access. What they're results showed is that you can record, while recording from V1, they did wide field imaging, and they show obviously the characteristic activity in V1, as well as some activity in somatosensory cortex next door, sort of more interior, in their view, where you get some activity because of potential whisking by the animal, and then a lot of activity in motor cortex related to the licking, but also their main result is that choice-related behaviors. So the earliest region of the brain that shows a distinction between trials where the mouse performed correctly and when the mouse performed incorrectly is found in secondary motor cortex. And like I said, they did monitor licking activity, whisking-related activity. So the take-home message is that in these rodent models, you are able to integrate, say, wide field imaging to capture more of the brain activity and account for how to explain larger cortical activity during these behavioral tasks in the rodents. And before I make this too much about motor behaviors, let's just go back to where else these art probes have been used in the visual system. Frontal eye fields is one. We do have an expert, and I'm not sure Matt mentioned, but we hope to have a special guest coming up in July. So if you're able to come back later in July, look for that, or if you're watching this, sometime in the future on YouTube, check back in our playlist of webinars to find the presentation on our probes being used in frontal eye fields. Likewise, superior colliculus. So I know folks that do this, and I did see a few publications that are currently in preprint. So just in case I did not present those here, not knowing where they were going to be published eventually, but they're coming. And I know this is more leaning toward oculomotor, which is fun for me, but for those of you most directly interested in vision itself, I did want to point out, very exciting, this week, the National Eye Institute at the National Institutes of Health published an article highlighting that the retina is a big target for people in the Brain Initiative to study just complicated circuits, right? So it's a very small prep, but so many different cell types, so many different interactions that I wanted to give you a taste for what it might be like to use NeuroNexus to record from the retina. So here's a quick example. You could customize a probe with a bunch of recording sites that cluster in, say, the ganglion cell layer, but have a stimulating electrode closer to the base of the retina. You could have your retinal prep. So we do recordings in vitro are totally fine. We can help design a way to hold your probe, position your probe, etc. Or if you wanted to record from an intact retina too, we could figure that one out. Retinal prep with your light underneath, your head stage over here. This is actually the 16-channel connector that would have been on that last probe, the polytrode probe that I showed you. Anyway, so NeuroNexus can help you record in the retina as well. So that's basically from the front to the back of the visual system.
All right, thanks a lot, Alexis. And a reminder that we're finally, after what, 3 months of having a webinar every week, we're going to take one off. So next week we will not have a webinar. So we're going to go ahead and end here. Thanks a lot for joining us, and see you in July with some later in July with some really great webinars. Thanks, everybody.
Thanks.